welcome to the Deep Dive. Today, we've got a pile of pretty technical source material all about biological agents.
That's right.
And our job is to cut through that, turn it into knowledge that actually makes sense and is, well, useful.
Yeah, because if you find all the talk about disease and infection a bit much.
Let's be honest, who doesn't?
Exactly. We're aiming to give you the fastest, most insightful path through it all.
So our mission today, as I see it, is really about pulling out what makes these biological hazards fundamentally different from, say, chemical ones.
Precisely. Because once you grasp their unique life cycles, it really does change how you have to think about risk assessment. Completely.
Okay, let's get started. We probably need a definition first. The sources mentioned the ILO, the International Labor Organization.
Yep, they give a pretty broad definition. A biological agent is basically any microorganism, cell culture, or human endoparasite, which may cause any infection, allergy, toxicity, or otherwise create a hazard to human health. It covers a lot.
Right, it's a wide net. So where are we focusing based on our material?
We're going to zero in on the 4 main types of microorganisms mentioned, and then crucially tackle the 4 special properties that make managing their risks so tricky.
Got it. So the 4 main types first. That's fungi, bacteria, viruses, and protozoa. Let's start with bacteria. They seem to be everywhere.
They really are. Simple, single-celled organisms. You find them in soil, water, even the deep ocean. But here's the kicker.
Go on.
The average human body. It actually contains about 10 times more bacterial cells than human cells.
That fact always gets me. We're basically walking bacteria colonies.
In a way. And luckily, most are harmless or even helpful, like our gut bacteria.
But not all of them.
No, definitely not. Some cause serious diseases like Legionnaire's disease. And the really tricky part for control, some bacteria, like Bacillus anthracis, that's anthrax, can form these incredibly tough spores.
Spores, right. Super resilient things.
Exactly. They can survive heat, cold, drying out. Makes them a real challenge to deal with.
Okay, spore formation is key there. Moving on to viruses. We talk about them all the time, but they aren't technically alive in the same way, are they?
Correct. Fundamentally, they're just genetic material, DNA or RNA, wrapped in a protein coat. Think of them as tiny little parasites, much smaller than bacteria or fungi.
And they need us or another host to replicate.
Absolutely. They have to invade a host cell and basically hijack its internal machinery to make more copies of themselves.
And that's what makes us sick. The hijacking process.
That process and our body's immune response trying to fight it off, that race between the virus replicating and our immune system responding is what causes the symptoms, like with a common cold.
And occupationally, what are the big ones?
Well, the sources really highlight bloodborne viruses, things like hepatitis B, hepatitis C, and HIV. They're a major risk in settings where there's exposure to body fluids, healthcare obviously, but other areas too.
Right. Okay. Next up, fungi. That includes molds and yeasts. We might think of like athlete's foot.
Yeah, that's a common surface infection, but the bigger occupational risk often comes from breathing them in. Fungi also reproduce using spores.
Spores again.
Yep. So imagine someone working with, say, moldy hay, like a farm worker. If they inhale huge numbers of Aspergillus spores.
That sounds bad.
It can be. It can cause serious lung diseases, like farmer's lung. It's all about the dose and the type of fungus, really.
Okay. And the last of the 4. Protozoa. Single-celled again, but different from bacteria.
They are single-celled, but the key difference is they do have a proper cell nucleus, which bacteria don't, and they're often mobile, they can move around.
And what's the big deal clinically?
The really crucial thing is that diseases caused by protozoa usually don't just go away on their own. Your immune system often struggles to clear them completely.
So you need treatment. Your body can't handle it solo.
Generally, yes. Medical intervention is usually required. We're talking about major global diseases here. Malaria, which is caused by Plasmodium.
Right.
Sleeping sickness, amoebic dysentery. These are serious protozoan infections.
Okay, so we've got our 4 players. Bacteria, viruses, fungi, protozoa. Now, let's get to that really critical point why these aren't just like chemical hazards, those 4 special properties.
Exactly, because, sometimes legislation lumps them in with hazardous substances like chemicals, but these properties make them fundamentally unpredictable in a way chemicals aren't.
The standard dose makes the poison idea doesn't quite sit.
It often breaks down completely, which leads us to property number 1, rapid multiplication.
Okay, how rapid are we talking?
Astonishingly rapid. The source material gives this example of E. coli bacteria. Under the right conditions, just one single cell
Yeah.
can multiply into over 200 trillion cells in only 24 hours.
200 trillion in a day.
Trillion. Let that sink in.
Okay, so if you thought a tiny, tiny initial exposure was safe.
That safety assessment becomes meaningless almost instantly. The hazard isn't static, it's growing exponentially inside the host. A microscopic exposure can overwhelm the body's defenses very quickly.
So the risk is dynamic, not fixed, like with a chemical dose.
Precisely. That's takeaway number one.
Right. Property number 2, rapid mutation. Why is that such a problem?
Well, think about it. If you're trying to control a risk, you want it to be predictable. But rapid mutation means the agent is constantly changing its genetic code. It's a moving target.
Making it harder for our bodies to fight it.
Exactly. The immune system might develop defenses against one version, but then the agent mutates, and those defenses might not work as well. HIV, for example, mutates very quickly.
And that complicates vaccines and treatments too, I imagine.
Hugely. It's a constant challenge to keep up with the changes.
Okay. Property 3, the incubation period. The delay between getting infected and actually feeling sick. This varies, right?
It varies wildly. I mean, dramatically. It could be just 1 to 3 days for the flu.
Pretty standard.
But then you look at something like Creutzfeldt-Jakob disease, CJD. The sources suggest a potential incubation period of 30 to 50 years.
Hold on, 50 years between exposure and symptoms.
Potentially, yes.
So, how on earth do you trace that back? If someone gets sick decades later, how do you pinpoint the workplace exposure that caused it? It seems almost impossible.
For immediate source tracing, it introduces enormous difficulty. You're right, it makes proving causation, handling compensation, and even knowing where to focus prevention incredibly hard after the fact.
Decades of uncertainty. Wow. Okay, property 4, infectiousness.
This is about spread. An infected person or animal can pass the agent on to others. Think of something highly infectious like TB, tuberculosis.
But not all biological agents are infectious person to person, right? Like Legionnaires.
Correct. Legionnaire's disease comes from the environment, not from another person. But when an agent is infectious, there's another layer of complexity. The asymptomatic carrier.
The person who's spreading it but doesn't know they're sick.
Exactly. They're infected, they're infectious, they might be shedding the agent, then they feel perfectly fine. Hepatitis C is often like this.
So you could have someone in the workplace unknowingly spreading the virus.
Yes, and that completely bypasses any health checks that rely on people reporting symptoms. It's a hidden risk.
Which leads us nicely into transmission pathways. The sources list 4 main ones: humans, animals, plants, and the environment. Let's focus on that human-animal link, zoonoses.
Right, zoonoses are diseases that jump from vertebrate animals to people. And certain jobs obviously carry a higher risk.
Like who?
Farm workers, vets, zoo workers, people working in sewage systems, abattoir workers, anyone with close contact with animals or their waste products.
Let's look at a couple of examples from the sources. First, cryptosporidiosis. What is that?
It's caused by a protozoan parasite. You usually get it from contact with infected animals like cattle or sheep, or maybe from contaminated water or food, often fecal contamination.
And the critical control point here.
This is vital. There's no vaccine and no specific treatment. Basically, you just have to manage symptoms like staying hydrated. So prevention is everything. It absolutely relies on meticulous hygiene, hand washing, clean water, proper sanitation.
Okay, no treatment, so hygiene is paramount. Now, contrast that with psittacosis, parrot fever.
Right, psittacosis. This one's a bacterial infection, usually caught from birds.
How do you catch it?
Typically by breathing in dust from dried bird droppings or respiratory secretions, often in areas where birds roost or are kept. Think attics, old buildings, pet shops.
But the key difference here compared to crypto.
Unlike cryptosporidiosis, this bacterial infection does respond to antibiotics. So while prevention is still important, if someone does get infected, there's an effective treatment available. Knowing the agent type protozoa versus bacteria changes the whole game plan.
That makes sense. And we can't discuss transmission without vectors, insects carrying disease. Malaria is the big one.
Absolutely. Malaria is caused by that Plasmodium protozoan we mentioned, and it's transmitted by the bite of an infected female Anopheles mosquito, usually at night. Still a massive global health problem.
And the incubation period here. You mentioned it can vary.
It can. Symptoms like fever, chills, headache might show up 7 to 18 days after the bite, but sometimes it can take up to a year or even longer before you get sick.
A year. So for people traveling to affected areas, the precautions need to be serious and ongoing.
Definitely. It's a package deal. Taking preventative antimalarial drugs, sleeping under insecticide-treated nets, using effective insect repellents, and crucially, being educated to recognize the early symptoms. Prompt treatment is key.
Okay, let's shift to some specific occupational examples and controls. Bloodborne viruses, HBV, HCV, HIV, you mentioned them earlier. Huge risk for healthcare workers.
It's often cited as the most common occupational disease risk for them. But worryingly, the sources also point to increasing risk for waste disposal workers.
Because in needles, sharps?
Exactly. Carelessly discarded needles and syringes and general waste pose a serious hazard. Now, interestingly, HIV itself is relatively fragile outside the body, low infectivity compared to hepatitis B, which is much tougher.
But the precautions are similar.
Yes, because the transmission route contact with infected blood or body fluids, often via a Sharps injury, is the same, so the core controls are procedural.
Meaning?
Things like using safer Sharps devices, ones with safety features built in, never recapping needles, immediate correct disposal in puncture-proof Sharps bins, and of course using appropriate PPE, like gloves, maybe eye protection, depending on the task.
Got it. Now, Legionella, the environmental one. It's in water and soil naturally, so how does it become a workplace risk?
The risk comes from inhaling airborne water droplets, aerosols that contain the bacteria. Think cooling towers, showers, spa pools, even decorative fountains if they aren't managed properly.
And the key to controlling it is?
Temperature. Legionella bacteria are quite fussy about temperature. They love lukewarm water, thriving between 20 and 45 degrees Celsius.
Okay, so what temperature range do we need to aim for in water systems?
You need to keep the hot water hot consistently above 60 degrees Celsius because that kills them. And keep the cold water cold, ideally below 20 degrees Celsius where they won't multiply readily.
So it's all about managing the water system itself.
Precisely. Appointing a competent person to manage the system, designing systems to avoid stagnant water, like keeping pipe runs short, using materials that don't provide nutrients for the bacteria, and that strict temperature control. You're basically engineering the environment to be hostile to them.
Makes sense. One more specific one. Leptospira bacteria, causing Weil's disease. Risk for sewer workers, farmers.
Yes, and anyone else who might come into contact with rats or contaminated water. The bacteria live in the kidneys of rats and other mammals and get into the environment through their urine.
How do people get infected?
Usually through contact with fresh rat urine or water contaminated with it. It can get in through cuts or grazes in the skin or through the eyes, nose, or mouth.
And the sources mention something really important about the symptoms and control here.
Yes, this is critical. The early stages of Weil's disease look a lot like flu. Fever, headache, muscle aches.
So someone might just think they have the flu and not seek help quickly enough.
Exactly. But Weil's disease progresses. Stage 2 can involve jaundice, kidney failure, and has a mortality rate of about 5 to 10% even with treatment. Time is absolutely critical.
Which brings us to that unique control measure.
The alert card. Workers who are potentially at risk, sewer workers, farmers, pest controllers, should carry a card.
What does the card do?
If they develop flu-like symptoms, they show the card immediately to their doctor. It tells the doctor about their occupational risk and prompts them to consider Weil's disease right from the start, ensuring faster diagnosis and treatment. It can literally save lives.
That's a really practical, vital control. Okay, so we've covered some very specific hazards and controls. If we zoom out, what are the general universal precautions for dealing with potential biological exposures?
Well, number one is always good basic hygiene. Proper hand-washing facilities, encouraging workers to use them frequently. Good welfare facilities are essential.
Makes sense. What else?
Sharp safety is universal, even outside healthcare. Proper handling and disposal. And a really simple but effective one, strictly prohibiting eating, drinking, smoking, or applying cosmetics in any area where contamination might be present.
Breaks that hand-to-mouth route.
Directly. And then, depending on the specific task, you might have other controls, like using remote cameras for things like sewer inspections to avoid entry, specific PPE for handling hazardous waste, or vaccinations were available, like for rabies for certain animal handlers.
Okay, that's a comprehensive look. We've covered a lot, the 4 types of agents, the critical differences from chemicals, specific diseases like Legionella and Weil's, and those targeted controls.
I think the absolute core takeaway, though, goes back to those special properties, that rapid multiplication, the mutation, the long incubation periods.
Yeah.
It means biological agents just don't follow that simple chemical dose-response relationship.
Right. That initial comparison to chemicals really falls apart when you look closely.
It does. Which
So,
brings us to the thought we want to leave you, the listener, with.
cool.
We started by saying these agents can be unlike chemicals. We highlighted that E. coli example, one cell becoming 200 trillion in a day. If you accept that multiplication potential, the inherent, almost explosive growth,
Yeah.
how does that force you to rethink safety? If a microscopic exposure isn't necessarily safe because the hazard itself can grow exponentially after the exposure, what does that do to your entire strategy? How do you plan for a risk that has its own life cycle, its own ability to amplify itself long after your initial control measures are done?
That's a really fundamental question. Rethinking safety based on the life cycle of the hazard itself. Lots to consider there.
Definitely food for thought.
Indeed. Thanks for tuning in to this deep dive.